that I'm muted for this. Okay, I think we are finally there. So I have got TikTok over here, I've got Instagram there, and I've got YouTube here. I can see it's not quite live on YouTube yet, but that could be the delay. Let's just see. I'm going to go for it and let's see what happens with YouTube. If it doesn't work today, I'll have to fix it for next time. So we're going through photosynthesis today, but specifically the light-dependent reactions. So the light-dependent reactions, we think about our chloroplast. You have your stacks of thylakoid membranes, which are also known as grana for plural or granum for singular. And you've got a double membrane, but it's the thylakoid membrane which is where the light-dependent reactions are happening. So that is the part that we're focusing on today. And there are three key stages for the light-dependent reactions. We have photolysis of water. We have photoionization of chlorophyll. And then we have chemiosmosis. So those are your three key stages. The main one that people find difficult is chemiosmosis. And also, I forgot to say at the start, can't believe I forgot to say this, but if any of my last year's year 12 are watching, I did promise that I'd do multiple shout outs throughout this year because I'm no longer teaching you because I'm not at school anymore. So a big shout out to who were my year 12s last year, who are now in year 13. And if you are watching, then yes, I'm still virtually your teacher this way. So hello if you are there. Also, for 2 hours only, I put a 50% discount code on my active recall workbook, which is something I'm going to be using throughout the live sessions on a Thursday. So if you want to get a copy of it, so when I'm using it in my lives, you can actually take part as well, then it's missestruck.co.uk. Use the discount code LIVE in capitals, and you can get 50% off the active recall workbook, either the entire course, the year 12 or the year 13 one, but that's only live for 2 hours at discount code. Okay, so back to photosynthesis, light-dependent reactions. We said they happen on the thylakoid membrane. We have photolysis of water, photoionization of chlorophyll, and chemiosmosis. But let's have a look at photolysis of water first of all. So the way to help you remember it is, whenever you see photo in biology, that means light. And whenever you see lysis, that means to split. So if you have photolysis of water, what's happening is inside of these thylakoid membranes, or embedded on thylakoid membranes, you have the pigment chlorophyll. And chlorophyll, if you remember from GCSE, it can absorb light energy. Chlorophyll absorbs that light energy, and that light energy then splits water. So that is the first step. We've got chlorophyll absorbs light energy, and in the exam you would have to be that specific and say chlorophyll absorbs the light energy for 1 mark for saying what photolysis of water is. Then you'd need to know what your products are. So this is like how we balance in biology. I know it's not proper balancing for chemistry, but half oxygen, and then you would have 2 electrons plus 2 protons. So that is what the water is split into. That oxygen is not used in photosynthesis. It is used in respiration though, so it could then go elsewhere in the plant, the mitochondria, and be used in respiration, but it's not going to be used in photosynthesis. So that is not going to come up again. But what is useful are the electrons and the protons that have been made in photolysis. The second stage we had here, which actually I say it's second stage, but it's happening simultaneously, that is your photoionization of chlorophyll. Again, we've said photo means light. Ionization, let's see how good your chemistry is. What does ionization mean? Thinking back to chemistry GCSE, I'm sure a lot of you do chemistry A level. Photoionization of chlorophyll. Even if you haven't learned this yet, you should be able to work it out because we've said photo means light, it's to do with light energy. Think about what ionization means, and then we've got the fact that it's happening to chlorophyll. So photoionization of chlorophyll is when light energy, again, is absorbed by chlorophyll. So the light energy is being absorbed by chlorophyll, but this time that energy is going to be causing the release of electrons from the chlorophyll. So it's been ionized because it's lost an electron. However, as we are all very aware, AQA are incredibly particular about the use of terminology in the mark scheme. So I'm not going to write the whole process here. What I'm going to write down is the key thing that AQA want you to say. Just like with photolysis of water, we had to say light energy absorbed by the chlorophyll, and then that energy is used to split the water. Same thing here, you get 1 mark for saying the light energy is absorbed by chlorophyll, but then you need to say that energy excites the electrons. So that is what they want you to say. The energy excites the electrons, and that causes electron to be lost. Okay, so we've now got to the point where process 1 results in the production of electrons and protons. Process 2 has resulted in the production of electrons as well. And these electrons and protons are now going to be used in this final stage, chemiosmosis, to make the 2 useful products. And they're useful because they're going to be used in, let me just move TikTok before my iPad falls off. They're going to be used in the light independent reactions. So that's why they are useful. And the 2 useful products that are created in chemiosmosis are ATP and, let's just go down here, reduce NADP. So those are the 2 useful products that we're going to get. So I'm going to quickly check Instagram can see okay. Yeah, all good. Instagram can see. I do have YouTube here now, and let's see. Oh yeah, lots of people managed to find me on YouTube. Excellent. And on TikTok. Awesome. So chemiosmosis, the 2 useful products are ATP and reduced NADP. If we think about the ATP first, there's a lot of overlap with biological molecules. In the light-dependent reactions, the energy for all of these processes is coming from the light. In the light-independent reactions, it's not coming from light, that's why it's light-independent, and it's actually coming from ATP. So ATP is going to be providing the energy for the light-independent reactions. So that's why ATP is going to be a useful product of chemiosmosis. It's going to be providing energy for the light-independent reactions. Reduced NADP, you might not have come across coenzymes yet. So let me just write that here. Now, coenzymes come up in respiration and in photosynthesis. It's not something you would have come across before, though, or quite likely haven't come across it before. Coenzymes are not catalysts, so it's not like enzymes as you know it. Coenzymes, they are going to be involved in the reaction. They're not used up themselves. They can be reused. But essentially, very briefly, what you need to know, surface level understanding of coenzymes for AQA, is that coenzyme is going to be picking up molecules and transferring them to another stage of the reaction. And for photosynthesis and respiration, it's picking up hydrogen atoms, which is a proton and an electron, and transferring it to a later stage. So that coenzyme, NADP, that is the coenzyme, but we create a reduced form of that. And it's reduced, this is where the chemistry comes into it. It's reduced because it's going to be picking up a hydrogen atom. So hydrogen atom. So this is our hydrogen atom. That is made up of, H plus is here, it's not that one. It's made up of a proton and an electron. So throughout photosynthesis and respiration, whenever you see a molecule gaining a hydrogen, I think oil rig is a gain of an electron. If it gains a hydrogen atom, that means it has gained an electron. And if it loses a hydrogen atom, it means it has lost an electron in that hydrogen. So reduced NADP is the coenzyme NADP, which had picked up a hydrogen. So sometimes you'll see it called reduced NADP. Sometimes you'll see it called NADPH, but it means the same thing. Because it's gained a hydrogen, it is now known as reduced NADP, and it's going to deliver that hydrogen and be used later on in the light independent reactions. Okay, so that takes us through photolysis of water, photoionization of chlorophyll, the products of chemiosmosis, and how those are going to be used in the light independent reactions. But we still haven't addressed in chemiosmosis how these electrons and protons are used to create these two products. And that is the final thing that we need to have a look at. And this is where I'm going to need to quickly wipe this board. So if you want to go and get a drink or something, I'm just going to be 1 minute. While I clear the deck, ready for the next diagram that we're going to need. While I'm cleaning this off, just as a reminder, you've got 2 hours until that discount code runs out. So that is an active recall workbook, which I'll show you. We're going to use right at the end of this session. So you can have a go at consolidating your knowledge. Okay, right. I'm going to have to consult my handy picture here because we're now going to do chemiosmosis, which involves me having to get creative and artistic and to try and draw a thylakoid membrane. So chemiosmosis. So this is happening on the thylakoid membrane. So I'm going to attempt to draw a phospholipid bilayer, because that is what your thylakoid membrane is. It's a phospholipid bilayer, and it has proteins embedded within it. Proteins. These are my phospholipids to make the bilayer. I'm just going to do one more. Then final one at the end, I'm going to do in orange. No idea why I just fancy it to be an orange. Right. This one, oh, orange might have been a bad idea. I don't know how I can see orange to try and get a bit more obvious. Here we go. So here is our thylakoid membrane. Let's get rid of that orange. It was rubbish. This is your thylakoid lumen. Thylakoid lumen. So this is, if I could do again those stacks of the thylakoids, the inside of one of those stacks is the thylakoid lumen. This line is representing the membrane, and this side of the membrane is the stroma. So that is what we're looking at in terms of location. And we have happening, I'm just going to quickly remind ourselves, we've got photolysis of water is happening. That is when water is split into oxygen, which doesn't get used again, protons, and the electrons. Those are picked up by this protein here. And this protein also contains chlorophyll. So let's colour it in green just to represent it contains chlorophyll. So this protein not only has the electrons from photolysis of water, it also has the electrons being released from photoionization of chlorophyll. So this protein contains those electrons. So that's as far as we've got so far. These electrons then move along what we call the electron transport chain, which is basically this chain of proteins embedded in the thylakoid membrane, the electrons can go from protein to protein. So that is our electron transport chain. And every time those electrons move along, they release a small amount of energy, and that energy is enough to pump a proton into the thylakoid lumen. So any of these protons, as the electrons move, they release energy, and that enables these protons to be pumped to the thylakoid lumen. So that means we end up with lots of protons, also known as hydrogen ions, in the thylakoid lumen. And because we've got lots of these protons on one side, we have a gradient, but we call it an electrochemical gradient because of that charge. So electrochemical gradient. So those electrons moved along the proteins in the electron transfer chain. That energy is used to pump protons to the thylakoid lumen. We now have lots of protons on one side of the membrane. That has created an electrochemical gradient. And if we think back to year 12, when you learned about transport across membranes, if you have something that is charged, it can't go through the phospholipid bilayer. It has to go through proteins by facilitated diffusion. And the only protein that the protons are complementary in shape and able to attach to is this orange one, which is ATP synthase, the enzyme that catalyzes the formation of ADP and PI into ATP. So as those protons move down their concentration gradient from the thylakoid lumen back into the stroma, it enables this enzyme, and you don't need to know the details of how it enables it, you just need to know it does enable this enzyme to cause the phosphorylation of ADP. So phosphorylation means adding a phosphate group. So ADP plus PI, and that is how we get ATP. So that's one part of chemiosmosis. It is a long process, this, but that is how the ATP is created. I'm just going to push TikTok back up. It's about to lose them. For some reason, my iPad holder is rubbish and always falls off. Okay, so that's how we made ATP. I'm going to very briefly say that once more. We had electrons released from photolysis of water, electrons released from photoionization of chlorophyll. These electrons move along the electron transport chain, which is proteins within the thylakoid membrane. As they move from protein to protein, it releases energy. That energy is used to pump the protons to the thylakoid lumen. We then have lots of protons on that side of the membrane, creating an electrochemical gradient. And because we've got an electrochemical gradient, the protons move down their concentration gradient, back into the stroma, through the only protein they can go through, which is ATP synthase. And that activates this enzyme to phosphorylate ADP to make ATP. So that's how we've made the ATP. The final thing, though, is we said you also get reduced NADP, which is the coenzyme that can pick up a hydrogen. And what is happening there, I go with my black pen, here's our NADP within the stroma. That NADP picks up the electrons from the end of the electron transfer chain, and it picks up the protons that have moved through ATP synthase, and an electron plus a proton is a hydrogen. And that is how we create our reduced NADP or NADPH. So it's picking up the electrons at the end of the electron transfer chain, it's picking up the proton, and together it makes reduced NADP. And that is chemiosmosis and the end of the light-dependent reactions. So it's showing you how we create reduced NADP and ATP, which are the two essential molecules needed for the light-independent reactions. Right, that is the light-dependent reactions. Let's have a look at some exam-style questions in my active recall workbook. For me to do this, I'm going to have to shift all the cameras over to the table now. So again, if you want to get a quick drink, this is going to take about 1 minute. So I need to go from the whiteboard. Or do I? Can I just zoom in? Oh, do you know what? I'm going to try zooming in. Let's see what happens. Get yourself a drink if you want, though, because this might take a while. It's a new style today compared to usual, and I'm working out as I go. So I'm going to stick my active recall workbook here and zoom in on all 3 devices on Instagram, YouTube, TikTok, and let's see what happens. I think we can probably guess. I'm probably going to mess something up, but we'll see. Right, so I'm going to need some sellotape. Oh, I should get blue tack really rather than sellotape. I'll do that next time. But for today, let's stick on here. Right, so this is one of the pages from my active recall workbook, which I'll say you've got 50% off for the next, I said 2 hours at the start, we're probably about an hour and a half in now, as in like left of the discount. So live is the discount code. The active recall workbook is like a stepping stone between writing notes, learning the theory, and doing exam questions. It's a step in between where it's lots of short answer questions and activities to test, have you understood the information? So that's what I wanted to do today, was to go through the information with the active recall workbook. Right, let's see. I said zoom in. Why don't I just go old school? Let's just move everything closer. Hope nothing falls over. We're getting closer. Okay, that looks all right. Oh, sorry, YouTube, you temporarily got shifted to an awkward position because I need to lengthen your cable. Right, yeah. YouTube, you've got the best view because I'm doing it on my actual SLR camera. So as long as that doesn't keep twisting, let's lengthen that some more. Okay, there we go. That should stop that camera twisting. Yeah, so YouTube, you can see okay. TikTok, I need to bring you over. Instagram's there already. And then I just need to squeeze myself in as well. Okay. Maybe even a bit of TikTok. I think we're good to go. Right, so this is just showing you the active recall workbook, and this is part of it for photosynthesis. And this bit here is chemiosmosis. I haven't actually got the pages here where it's linking to photolysis and photoionization of chlorophyll, but this is like one example. So we've got to use a diagram to describe chemiosmosis in 4 stages. So what I just drew there, we've now got here to think about. How could we summarize that in 4 key stages? So I'm going to say electrons from photolysis and photoionization move along the electron transport chain and release energy to pump protons into these, not strains, sorry, thylakoid membrane. I don't know how well you can see this, and even if you could see it, I have the most appalling handwriting when I'm using a biro, and I'm doing it on a whiteboard as well, so it probably just looks like a load of scrawls on paper. But I'm narrating it as well, so hopefully you can read it. The next thing then, I'm going to say energy. Oh no, we've already said that energy is used to pump the protons. I'm going to say creates an electrochemical gradient. and H plus moves through ATP synthase to make ATP. And then, let's see if I can get my YouTube camera to focus a bit better on this. Then I'm going to say electrons and H plus picked up by NADP to become reduced NADP. I actually could have split that out further. I said 4 key steps and I've squashed it all into those. Right, then we've got some short answer questions. We haven't actually done the light independent reactions today, so we can't do that one. There was another page as well here, which is linked to the light independent reactions and also limiting factors. But again, that is something that we haven't focused on today. Maybe we could do that next week, or I take requests on based on what you'd like to do. But that's just showing you a little bit then of the active recall workbook. I'm going to move you all back just to finish off. There we go. All right, let me just check what everyone can see before I sign off for this evening. I think we can all see me. Yes, okay. So that is the first one of the new style of weekly lives. Last year, or the last 2 years, it was just doing exam questions. But I know a lot of people want help, they want tuition. I don't have any space on my tuition. Tuition is very expensive as well, so people are more interested in having some kind of free tuition, not just exam questions, which is why this year is going to be a mix of, I will be teaching you the content or reviewing the content. You can request which topics you want by commenting on the recordings each week. So the recordings will be saved on my Instagram and on my YouTube channel. Both of those are Miss Estruch Biology. If you comment on the recording and say what you'd like next week, whichever is the most common request, that's what I'll be doing. And I'm going to be avoiding repeating topics so we can ideally go through as much of the A-level as possible. And if there's no obvious request, I'm just going to be teaching what I know you'll be probably learning at school at that time. So that is what I will be doing. You can see that YouTube can't actually see me. Oh, well, it doesn't really matter. And then we'll do some of the active recall workbook each time and look at key marking points. So that's what I'll say, if you do want to get the active recall workbook, so you can actually take part each time doing the questions, that is why I'm giving 50% off for the next 2 hours. No idea what the time is now, though, let's see. But oh, it's only 8:30, 25 to 9, I thought it'd be later. So you've got an hour and a half left on that discount. Hopefully you found it helpful. I know there was a few teething issues at the start, so I couldn't get YouTube to work, but hopefully that will streamline and work next time. I'll try and work out as well whether it's better for me just to pin up here or to just do it zoomed in on the table, because then you'll be able to see this more clearly and I'll be able to write more neatly. But any feedback at all that's going to be helpful to make sure these sessions are as useful as possible, then send me a message on Instagram or leave a comment on the recordings so I know which bits you found particularly helpful. I can try and focus on those more. And if there were any technical issues that I wasn't aware of, like sound or anything, then let me know. But that's it for this evening. And it was so great to see so many people here. So I think I had at least 50 on Instagram, between 50 and 100 on TikTok, and YouTube. I think there were between 50 and 100 there. So it's quite a good group of you for the first session. Best of luck. And as I said, if any of my year 12s from last year are watching, I hope you enjoyed that dedicated session to you, and I'll see you all next week.